Hello, and thank you so much for taking the time to look at our poster. My name is Avery Williams, and I'm an undergraduate research assistant in the Lassane Lab in the Department of Cell, Developmental, and Integrative Biology, which is in the School of Medicine at the University of Alabama at Birmingham. Today, I would like to discuss with you my lab's efforts in optimizing model selection for glioblastoma utilizing gene expression. There is always room for improvement in disease treatment, and one of these areas is through developing better preclinical models. With an effective preclinical model, we can study how a treatment might interact with and hopefully mitigate a certain disease and have a good idea of its viability before having to introduce it to people in clinical trials. In this case, we chose to focus on cell lines and patient-derived xenograft mouse models. If you aren't familiar with the latter, the mouse patient-derived xenograft, or PDX, involves implanting patient tumor tissue in immunocompromised mice. The disease we chose to focus on is glioblastoma, which, as you can see in the introduction, affects around 210,000 people worldwide a year. This disease model is particularly useful here because glioblastoma is a stage 4 brain cancer that involves aggressive tumors, which are more easy to establish in PDX models. Further, it is a debilitating disease that currently has very low survival and high relapse rates, providing it with a particular exigency for effective treatments. So while our focus here is with these two models and this particular disease, our long-term goal, as shown in this objective section, is to be able to better match a patient with an effective model and develop computational approaches to assess and analyze model efficacy. From here, we can move into our methodology. Unfortunately, due to lack of availability from any single source, our patient tissue and model samples did have to come from 3 different public data consortia, which are the Cancer Genome Atlas, the source of our patient tissue samples, the Cancer Cell Line Encyclopedia, which, as the name entails, provides cell line samples, and the Mayo Clinic's Brain Tumor Patient-Derived Xenograft National Resource, which is the source of our PDX mice samples. To combat batch effects, we pulled the data from a resource called Recount3, which uniformly processes raw RNA-seq data with the monorail system to make cross-comparison between consortia more accurate. From here, we ran the data through the DE-seq2 program, which provided detailed results as to differentially expressed genes between the patient tissue and each of the preclinical models. We then ran analyses such as the Pearson correlations depicted in figures 3 and 4 to visualize the correlation between our sample groups, and were able to expand our analyses to seek global patterns that could suggest advantages and weaknesses for each of our models. Moving to the discussion section, we were able to rank the differential expression of over 60,000 genes by the p-values and log2 fold changes computed by DE-seq2. From a shortened list of the genes more significantly differentially expressed, we sought out genes that might be related to glioblastoma etiology and progression, as these genes are important to accurately recapitulate expression in preclinical models of the disease. An example of expression performance for one of these genes is BCAN, which is depicted in Figure 1, and codes for a protein that has been studied to be highly expressed in gliomas and may play a role in the growth and cell motility of brain tumor cells. We can see from this figure that while the PDX models seem to model patient tissue expression of the gene relatively well, the cell lines fell significantly short and had very low counts for this gene. This pattern was shown in a large portion of these genes studied, also including the genes KCNJ10, OLIG1, and others. Further, we noticed that many of the genes on our list of significantly differentially expressed genes were related to the immune system and cell signaling. An example of this is the CD163 gene, which is depicted in Figure 2 and is expressed in monocytes and macrophages. As you can see in the figure, while the patient tissue showed a broad range of expression after normalization that was concentrated relatively low to begin with, the models showed next to 0 expression levels. This is as expected when considering the biology here, as cell lines do not have immune systems, and the PDX mice have been immunocompromised, and cell signaling could diminish when cells are in a model format. Other than looking at smaller scale significant differences, we could visualize global differences through the Pearson correlations, which is shown here in figures 3 and 4, as well as p-value and log2 fold change that were previously mentioned. It is of note that the cell lines had a lower overall p-value, both mean and median, the latter of which minimizes the effect of outliers, when compared with patient tissue counts than PDX mice did. And here's a table of that. So, to begin to conclude, while further analysis of this data would be beneficial, the use of differential expression analysis here and elsewhere has been shown to be advantageous in determining possible weaknesses in models. These weaknesses could be seen as possible areas for improvement or taken into consideration when determining the best model for a certain circumstance. In the future, we plan to continue this analysis by introducing data we generated from additional PDX models and by expanding out to include patient blood samples in our comparison. Overall, it is vital that we continue to strive to improve the accuracy of preclinical models in order to realistically recapitulate patient disease etiology and progression in order to better test possible treatments. I would like to finish by thanking the members of my lab and the American Cancer Society for funding this research. For any questions, comments, or concerns, you can contact my PI, Dr. Brittany Lassane, at bnp0001@uab.edu. Thank you so much.